So hey guys, today we are starting a biology chapter, respiration and circulation. It is on page number 153 of the textbook. So let's get started. All living organisms require energy to carry out various life processes. The energy that is stored in the body in the form of complex organic compounds is however not usable by the organism unless it is converted into usable form. This conversion is achieved through the process of respiration. Now let's see what is respiration. Respiration, it is a biochemical process of oxidation of organic compounds in an orderly manner for the liberation of chemical energy in the form of ATP. So here it is written C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus 38 ATP. C6H12O6 is glucose, O2 is oxygen, CO2 is carbon dioxide, H2O is water, and ATP is adenosine triphosphate, which is energy. For this, the process of gaseous exchange takes place between the organism and the environment. The site of gaseous exchange is called the respiratory surface. Now let's see organs of respiratory exchange. Respiratory exchange is a simple physical process for efficient gaseous exchange. The respiratory surface should have the following features: A. It should have a large surface area. B. It should be thin, highly vascular, and permeable to allow exchange of gases. C. It should be moist. Gaseous exchange in plants. The shape and structure of plants facilitate gaseous exchange by diffusion. A terrestrial flowering plant has many air spaces between the cells of stem, leaf, and root. These air spaces are continuous. Oxygen diffuses into the air space through stomata. Stomata is the pores on leaves and young stems. Carbon dioxide and water vapor diffuse out. In the aerated soil, the oxygen dissolves in the film of moisture or water around the root tissue and enters it by diffusion. Woody flowering plants, trees, and shrubs have an external impervious bark. Here, gaseous exchange occurs through small pores in the stem surface called lenticles. Now, let's see respiration in animals. As compared to plants, animals show wide variety of respiratory surfaces or organs. The respiratory surfaces differ in various animals. In animals, depending upon the complexity of organization and the surrounding medium, certain parts of the body have become specialized into different types of respiratory organs. In the higher animals, these respiratory organs are also associated with a transport system. Now see the table given: organisms, their habitat, and respiratory surface or organ. So protists, sponges, and coelentrates are aquatic animals, organisms, and they respire through plasma membrane. Then we have flatworms like planaria, annelids, amphibians, etc. These are aquatic or semi-aquatic. They respire through plasma membrane. General body surface is moist skin. Insects are terrestrial organisms. They respire from tracheal tubes and spiracles. Arachnids like spiders and scorpions are terrestrial organisms and they respire through book lungs. Limulus are aquatic and they respire through book gills. Amphibians like frogs, salamanders, and newts are aquatic and they respire through external gills. Fish are aquatic and they respire through internal gills. Reptiles, birds, and mammals are terrestrial organisms and they respire through lungs. Turtles are underwater organisms and they respire through cloaca. Now let's see human respiratory system. The respiratory system brings about inspiration, expiration, and exchange of gases in the lung. These are then transported by blood from the lungs to the different tissues and part of the body. The respiratory system can be divided into an upper respiratory system having external nares, nasal cavities, internal nares, nasopharynx, nose, throat, and associated structures. The lower respiratory system refers to the larynx, trachea, bronchi, bronchioles, and lungs. Now let's see the diagram. So this is the diagram that you can refer for the various organs or various parts of the human respiratory system. You can see nasal cavity, nostril, mouth, pharynx, larynx, trachea, right main bronchus, left lung, right lung, ribs, diaphragm, alveolus, air sac with blood capillaries, etc. Now let's see the first respiratory organ, that is nose. The nose has a pair of slit-like opening called external nares or nostrils for entry of air into the nasal cavity. The nasal cavity is divisible into right and left nasal chambers by a mesenteroid cartilage. Each nasal chamber is further divided into three regions: vestibule, respiratory part, olfactory or sensory chamber. Vestibule, it is the proximal part about the nostril. Its skin has hair for filtering the air and trapping the dust and suspended particles in the inhaled air. Respiratory part, it is a conditioner. The middle thin walled, highly vascular part for warming and moistening the inhaled air. Olfactory or sensory chamber, the uppermost part is lined by olfactory epithelium for detection of smell. So vestibule, it is for trapping the dust particles. Respiratory part is for conditioning and moisturizing the air. Olfactory is for detecting the smell. Then come to next part, that is pharynx. It is divisible into three parts. The nasopharynx, it is the uppermost part. The nasal chamber, from the nasal chamber it leads to oropharynx. The oropharynx is the common passage for food and air. This continues below as the laryngopharynx. Between the naso and oropharynx is the palate bone. The pharynx has a set of lymphoid organs called tonsils. Now let's go to larynx. It is called voice box. It is the part of the respiratory tract which contains vocal cords for producing sound. The larynx extends from the laryngopharynx in the hyoid bone to the trachea. It is a hollow tubular structure. Its wall is made up of cartilage plates held by membranes and muscles. Internally, it is lined by a pair of folds of elastic vocal cords that are true vocal cords. Voice is produced by passage of air between the vocal cords and modulations created by tongue, teeth, lips and nasal cavity. The larynx open into the laryngopharynx through a slit-like opening called glottis. This opening of the trachea or windpipe is guarded by a leaf-like flap called epiglottis. It prevents the entry of food into the trachea. So this is the diagram of larynx. You can see the epiglottis that prevents the entry of food into the trachea. Then we have hyoid bone, thyrohyoid membrane, laryngeal prominence, Adam's apple. Men have Adam's apple. Then we have thyroid cartilage, tracheal cartilage. Now let's move to trachea, that is windpipe. It is a long tube 10 to 12 centimeter in length. It runs through the neck in front of esophagus and extends into and up to the middle of thoracic cavity. It is supported by C-shaped 16 to 20 rings of cartilage which prevents the collapse of trachea. It is lined internally with ciliated pseudostratified epithelium and mucus glands that trap the unwanted particles, preventing their entry into the lungs. Then we have bronchi. The trachea divides into right and left primary bronchi as it reaches the middle of the thoracic cavity. The bronchi are supported internally by C-shaped incomplete ring of cartilage. The primary bronchi divides to form secondary and tertiary bronchi, which leads into terminal bronchioles ending into alveoli. So we have branches, the left branch, right branch, primary bronchi, and then bronchi get into bronchioles, and then bronchioles into alveoli. Lungs, these are the main respiratory organ of human. One pair of spongy and elastic lungs are present in the thoracic cavity. Each lung is enclosed and protected by a double pleural membrane, outer parietal and inner visceral membrane. Between the two pleura is a pleural cavity filled with a lubricating fluid called pleural fluid. It is secreted by the membranes. The right lung is larger and is divided into three lobes, while the left lung is smaller and is divided into two lobes. Each lobe of the lung has the terminal bronchioles ending in a bunch of air sacs, each with 10 to 12 alveoli. Now let's see alveoli. These are thin walled lobulated structures like a bunch of grapes. Each alveolus is surrounded by a network of capillaries of pulmonary arteries and veins. These have highly elastic wall made up of a single layer of squamous epithelium resting on a basement membrane of connective tissue. These are about 700 million. There are about 700 million of alveoli in the lung, and they provide the surface area for exchange of gases. Now let's see diaphragm. It is a muscular septum that separates the thoracic and abdominal cavity. It is dome shaped, and on contraction it becomes flattened. Mechanism of respiration. Respiration is a biological process involving exchange of gases between the atmosphere and the lungs, and it results in the formation of ATP. It includes the following processes: A. Breathing, B. External respiration, C. Internal respiration, and D. Cellular respiration. Now let's see the diagram. So here is the diagram of breathing. When we inspire, means when we breathe in, what happens is the lungs expand, matlab the because of the pressure the lungs expand and the diaphragm contracts and moves down because of the air filled in the lungs. And now when we expire, means during expiration, breathing out, what happens? The rib cage moves down and inward, means the lung contracts and the diaphragm also relaxes and moves up. Now let's see breathing. It is a physical process by which gaseous exchange takes place between the atmosphere and the lungs. It involves inspiration and expiration. Both these steps involved parts of the thoracic cage, the ribs, sternum, and the intercostal muscles and muscles of the diaphragm. Inspiration. During inspiration, the atmospheric air is taken into the lungs. It occurs due to the pressure gradient formed between the lung and the atmosphere. It is an active process in which the diaphragm becomes flat and goes downward. The external intercostal muscles contract, so the ribs and sternum moves upward and outward. This leads to an increase in the thoracic volume and decrease in pressure of thorax and the lungs. To equalize the low pressure inside the lung, air from the atmosphere rushes into the lung. This is inspiration. Now let's see expiration. During expiration, the thorax contracts, causing air to be exhaled. The diaphragm relaxes and is pushed upward. It becomes dome shaped. The intercostal muscles also relax, pulling the rib cage inward and downward. This causes a decrease in thoracic volume and leads to increase in pressure in the thorax and the lung as compared to the atmospheric pressure. So air from the lung rushes out. This is expiration. One inspiration and one expiration is one breathe. Now let's see B, that is external respiration or exchange of gases at the alveolar level. An alveolus consists of a layer of simple squamous epithelium resting on a basement membrane. It is intimately associated with a dense network of capillaries. The capillary wall is also made up of simple squamous epithelium resting on a thin basement membrane. Both the layer have similar structure and are thin walled. Together they make up the respiratory membrane through which gaseous exchange occur, that is between the alveolar air and the blood. Diffusion of gases will take place from an area of higher partial pressure to an area of lower partial pressure until the partial pressure into the two region reaches equilibrium. The partial pressure of carbon dioxide of blood entering the pulmonary capillary is 45 mmHg, while partial pressure of carbon dioxide in alveolar air is 40 mmHg. Due to this difference, carbon dioxide diffuses from the capillary into the alveolus, that is from the high pressure to the low pressure. Similarly, partial pressure of oxygen of blood is in pulmonary capillary is 40 mmHg, while in alveolar blood it is 104 mmHg. Due to this difference, oxygen diffuses from alveoli to the capillaries. So here this is the diagram of exchange of gases between alveoli and capillary. You can see the diagram carefully. It is very important for board exams. Then we have pulmonary volumes and capacities. Okay. So lung values, and we have also lung capacities. So tidal volume. Tidal volume TV, it is the volume of air inspired or expired during normal breathing. It is 500 ml. So it is normal breathing and inhaling and exhaling. Then we have inspiratory reserve volume, that is IRV. The maximum volume of air or the extra volume of air that is inspired during forced breathing is in addition to TV, its volume is 2000 to 3000 ml. Extra volume of air that we inspire while breathing is the inspiratory reserve volume, and opposite to that is expiratory reserve volume, that is ERV. The maximum volume of air that we exhale or expire during forced breathing after normal expiration, its volume is 1000 to 1100 milliliter. Then we have dead space. The volume of air that is present in the respiratory tract from nose to the terminal bronchioles but not involved in gaseous exchange, it is 150 ml. So 150 ml of air is always present in our respiratory tract. It does not participate in exchange of gases. So that is dead space. Then we have residual volume, the volume of air that remains in the lung and the dead space even after maximum expiration. It is 1100 to 1200 milliliter. So that remains in the lung even after we forceful expiration also, we cannot take the air out. So that is residual volume. Then we have lung capacities. Total lung capacity, the maximum amount of air that the lung can hold after a maximum forceful inspiration is 5200 to 5800 milliliter. इससे ज़्यादा the lung cannot hold the air. Then we have vital capacity, the maximum amount of air that can be breathed out after maximum inspiration. It is the sum total of TV, IRV and ERV. So ये तीनों का addition, TV, IRV and ERV का addition is the vital capacity, that is 4100 to 4600 milliliter. Now let's see internal respiration. The two main components of blood involved in transport of the respiratory gas CO2 and O2 are the RBCs and the plasma. Number 1, transport of oxygen. Of the total oxygen transported, only 3% is transported in a dissolved state by the plasma. The remaining 97% is bound to the hemoglobin present in the RBCs. Hemoglobin acts as the respiratory carrier. It has a high affinity for O2 and combines with it to form oxyhemoglobin. So oxygen and hemoglobin are true partners. They cannot live without each other. So they form a compound, molecule, that is oxyhemoglobin. Theoretically, one molecule of Hb has 4 Fe++, each of which can pick up a molecule of oxygen. So here is the reaction. Hemoglobin plus 4O2 gives hemoglobin 4O2. Oxyhemoglobin is transported from lungs to the tissue where it readily dissociates to release O2. Hb now is getting dissociated into Hb plus 4O2. However, the degree of saturation of hemoglobin with oxygen depends upon the oxygen tension, that is PPO2. Oxygen tension. 100% saturation is rare. Maximum saturation of 95 to 97 is at PPO2 in alveoli, 100 mmHg. Degree of saturation decreases with the drop in PPO2. Partial pressure of O2. This begins the dissociation of HbO2. At 30 mmHg of PPO2, only 50% saturation can be maintained. The relationship between HbO2 saturation and oxygen tension is called oxygen dissociation curve. This oxygen hemoglobin dissociation curve is a sigmoid curve, and it shifts toward the right due to increase in H plus concentration, increase in PpCO2, and rise in temperature and rise in DPG. 2,3 diphosphoglycerate formed in the RBC during glycolysis. It lowers the affinity of hemoglobin for oxygen. Now we'll see Bohr's effect. It is the shift of oxyhemoglobin dissociation curve due to change in partial pressure of CO2 in blood. So this is Bohr effect. It is very simple. Just it is shift of the sigmoid curve due to due to change in partial pressure of CO2. Haldane effect. Oxyhemoglobin functions as an acid. It decreases pH of blood due to increase in the number of H plus ions. HCO3 minus changes into H2O plus CO2. In the alveoli, where PPO2 is high and PpCO2 is low, oxygen bind with hemoglobin, but in the tissue where PPO2 is lower and PpCO2 is high, hemoglobin does not hold as much O2. It releases O2 for diffusion into the tissue cells. So this is the Bohr effect curve. It just shifts into the right because of the partial pressure of CO2 in blood. Now we'll see transport of CO2. Yeah. So these boxes are actually not evaluative in your exam, so I'm not reading these ones. I'm only reading the text. Transport of CO2. Carbon dioxide is readily soluble in water and is transported by RBCs and plasma in three different forms. By plasma in solution form, 7% only 7% of CO2 is transported in a dissolved form as carbonic acid, which can break down into CO2 and H2O. You can see the reaction here. Then we have bicarbonate ions. 70% of the carbon dioxide is dissociated by, if here it is written that it is only dissociated by dissolved in plasma by 7%, while here it is 70%. Nearly 70% of carbon dioxide released by the tissue cells diffuses into the plasma and then into the RBCs. In the RBCs, CO2 combines with water in the presence of a zinc containing enzyme, carbonic anhydrase, to form carbonic acid. Carbonic anhydrase enzyme is found in the RBC and not in the plasma. The rate of formation of carbonic acid inside the RBC is very high as compared to its formation in the plasma. Carbonic acid, being unstable, almost immediately dissociate into the HCO3 and H plus ion in the presence of enzyme carbonic anhydrase, leading to large accumulation of HCO3 inside the RBC. In thus moves out of the RBC. This would bring about imbalance of charge inside the RBCs. To maintain the ionic balance between the RBCs and the plasma, chlorine diffuses into the RBCs. This movement of chlorine ion is known as chloride shift or Hamburger's phenomenon. HCO3 that comes into the plasma joins to Na plus and K plus, forming NaHCO3 or KHCO3 to maintain pH of blood. H plus is taken up by protein hemoglobin. You can see the reactions. I am not reading out the reactions. You can just learn the reactions. These H plus ions might be expected to lower blood pH, but they are buffered by hemoglobin by the formation of deoxyhemoglobin at the level of lungs in response to the low partial pressure of carbon dioxide of the alveolar air. Hydrogen ion, here is the diagram you can see. So yeah, you can see these diagrams. Hydrogen ion and bicarbonate ion recombine to form carbonic acid, and under the influence of carbonic anhydrase yield carbon dioxide. By red blood cell it is 23%. Carbon dioxide binds with the amino group of hemoglobin and forms a loosely bound compound, carboamino hemoglobin. You can also say that this molecule readily decomposes in region where the partial pressure of carbon dioxide is low, releasing the carbon dioxide. We'll see last part, that is cellular respiration. I think it is the last. It is the last step taking place inside the cell where food is oxidized and ATP is generated. It can be shown by two steps: oxidation and phosphorylation. Oxidation, breaking down of complex organic molecules into simple inorganic molecules with release of heat energy. Then phosphorylation, it involves trapping the heat energy in the form of high energy bond of ATP molecules. ATP is used to carry out vital life processes and so is called as energy currency of the cell. Then we have regulation of breathing. Respiration is under dual control, nervous and chemical. Human adults breathe about 12 times per minute, while a newborn about 44 times per minute. Normal breathing is an involuntary process. Steady reading of respiration is controlled by neural located in the pons and medulla and are known as the respiratory centers. It regulates the rate and depth of breathing. It is divided into three groups: dorsal group of neurons in the medulla, ventral lateral group of neurons in the medulla, and pneumotaxic center located in pons. Slow wave primarily limits inspiration, slow wave sleep, and rapid eye movement sleep. Apneustic center in the medulla is antagonistic in the neuromotaxic center. It controls non-rapid eye movement sleep and wakefulness. You can see the diagram. Regulation of breathing. During inspiration, when the lungs expand to a critical point, the stretch receptors are stimulated and impulses are sent along the vagus nervus to the expiratory center. It then sends out inhibitory impulses to the respiratory center. The inspiration muscle relax and expiration follows. As air leaves the lungs during expiration, the lungs are deflated and the stretch receptors are no longer stimulated. Thus, the inspiratory center is no longer inhibited and a new respiration begins. This event are called the Herring-Brewer reflex. The Herring-Brewer reflex controls the depth and rhythm of respiration. It also prevents the lung from inflating to the point of bursting. The respiratory center has connections with the central cerebellum cortex, which means we can voluntarily change our pattern of breathing. Voluntary control is protective because it enables us to prevent water or irritating gases from entering the lungs, but the ability to stop breathing is also limited by the buildup of carbon dioxide in the blood. Modified respiratory movements. This part I think is deleted, so we'll not read this part. We have common disorder of respiratory system. The given table show a list of common respiratory disorders, their symptoms, cause and treatment. So these are the disorders, their symptoms, cause and treatment. You can just read this. Very easy. It is emphysema, chronic bronchitis, acute bronchitis, sinusitis, laryngitis, pneumonia, asthma, and we have occupational respiratory disorder, silicosis and asbestosis. You can read these. Then we have artificial ventilation. This is also deleted from the syllabus. Now we'll move in transportation in living organisms. All living organisms, whether unicellular or multicellular, show an important property of exchange of material with their surrounding as well as between various parts of their cell or body. Organisms take up oxygen and nutrients from the surrounding. These are circulated within the body for various metabolic activities. The waste generated within are given out into the surrounding. Transportation is in organisms and animals occur by diffusion and by active transport between the cell. This mechanism is suitable where the surface area of body is large and the distance between parts of the body in the organism is extremely small. Cyclosis is the streaming movement of the cytoplasm shown by almost all living organisms. Example: paramecium, amoeba, root hair cells of many plants, and WBCs in animals. It is for transportation within the cell or intracellular transport. In sponges and coelentrates, the surrounding water is circulated through the body cavities. In flatworms, where there is parenchymal circulation, in roundworms there are no blood vessels and the body fluid is moved around the viscera by contraction of body wall and muscle. This is extracellular transport rotation, and here after this we have circulation, which we will continue in the next part. So stay tuned for the next part, and thanks for watching. Please like, share and subscribe.